Hello, I'm Betty, a senior embryologist for 25 years and the clinical director of BAIBYS.
And I'm Yaron, CEO of BAIBYS. We thank the organizer for their kind invitation to present here, and we are very happy to present the BAIBYS system for you.
So let's start. When the ICSI method was implemented in the IVF lab, the impression was that the male factor was no longer an issue. Is this indeed the fact? Every sperm sample contains a huge variety of sperm cells quality, millions of options for fertilizing the oocyte and creating an embryo. But in fact, very few of the sperm cells can actually be used for ICSI, since the sperm DNA integrity plays a critical role in creating a good enough embryo to result in live and healthy baby. Even in the worst samples, few normal sperm cells usually exist. The challenge is to locate them and isolate them for fertilization via ICSI. Thinking of this, the sperm cell is the only cell that can actually be selected for a more successful IVF cycle. Oocytes are a given fact, and the embryos obtained are already the next step of the process that can only be prioritized for transfer or freeze. In reproductive physiology, many abnormalities may occur during spermatogenesis, spermiogenesis, epigenesis, resulting in spermatozoa defects. These disorders may concern sperm morphology, numerical or structural chromosome abnormalities, abnormal chromatin, sperm DNA defects, and these result in oocyte activation failure, no fertilization or zygote blocking, chaotic early embryo development, negative blastocyst culture, miscarriage, and birth defects. And oocytes seem to be able to repair some of the sperm DNA defects, but not all. Oocytes of women above the age of 38 show diminished repair capability. The average age of women undergoing IVF increased in the modern world. Therefore, choosing the best sperm becomes even more critical. So on the left side, we can see what considered by the WHO a normal sperm cell, which is symmetric, proportional, and no morphological defects. On the contrast, we can see on the right-hand side an abnormal sperm cell, which is asymmetric, non-proportional, has morphological defects like vacuoles or others. Upon fertilization, a normal sperm cell can result in good quality blastocysts, but an abnormal sperm cell will almost never produce a good quality blastocyst. A sperm cell cannot be tested for its DNA integrity directly and then be used for fertilization. Therefore, sperm morphology and motility, which are highly correlated with its DNA quality, are the only direct parameters that can be evaluated on a live sperm without harming it, and that can be used for more accurate selection before fertilization. High magnification microscopy allows revealing defined morphological defects that cannot be revealed during standard ICSI magnification and have a clinical impact on the developing embryo and the IVF outcome. Recent studies demonstrated strong tendency between selection of sperm in high magnification and its clinical outcome. Selected sperm cells with normal morphology in high magnification for fertilization were correlated with reduced structural defects in developing embryos in compared to regular ICSI, significantly reduced rates of birth defects often associated with ICSI, and lower DNA methylation levels with higher gene expression of many critical genes related to normal embryo development. Casuta et al. Study published in 2023 confirmed the link between different omics patterns like epigenome, methylenome, transcriptome, and microRNA with sperm head morphology in high magnification. This study results suggested that microRNA profiling of good morphologically sperm may provide biomarkers reflecting sperm function and quality. This innovative approach opened a new perspective to explore male infertility, in addition to the basic semen analysis as published in the 6th edition of the WHO, using new diagnostic tests, particularly during the actual ART procedure. Another study demonstrated the difference in the global microRNA expression profiles of sperm samples with good and poor morphology. This study found a significant difference in low versus normal microRNA expression levels at 10 different samples scored according to their high magnification morphology. You can see the normal in red and the abnormal in blue, dividing the two populations into two distinct groups based on their gene expression profiles. Another very important link between sperm morphology and gene expression was found by exploring a group of top-ranked functional protein interaction network in the sperm cell, which is known to be critical for early embryo development. The gene expression level of these proteins was evaluated in normal and abnormal sperm selected in high magnification. The descriptive results show a general trend of underexpression in 10 top-ranked genes in morphologically abnormal sperm cells. These results provide additional important link between sperm morphology as seen using high magnification and the DNA integrity and functionality of such a cell. Another study I decided to present to you here compared intratelomere distance, ITD, and chromosomal territory area, CTA, of chromosome number 1, which is the largest chromosome, therefore was the model here, between subjects with high normal and low abnormal sperm head morphology scores using FISH method. Both the ITD and the CTA were found to be significantly higher in subjects with abnormal sperm morphology versus normal sperm morphology in high magnification, indicating possibly improper DNA condensation. As we all know, proper DNA condensation in the sperm cell is critical for its DNA protection and function. And the conclusions of this study were a link between nuclear architecture and sperm head abnormalities, particularly vacuoles that can only be seen in high magnification. And selecting spermatozoa with normal head morphology leads to better nuclear architecture using high magnification might indirectly explain the positive ART outcomes observed with this technique. So we all understand the importance of using better sperm selection before ICSI. So where are still the limitations? As of today, using high magnification sperm selection before ICSI did not become a gold standard in the IVF routine because it was currently be performed only manually. And therefore, it is very time consuming, about 3 hours to collect 15 normal sperm cells, and most IVF centers do not have enough trained manpower even for more basic procedures. It's a global problem. So how can we focus on improving IVF results? It is inconsistent and subjective because it is very much depending on the human factor as the embryologist experience, tiredness, workload, personal stress, distracting emotions, and more. It requires extensive training before clinical implementation, and therefore it is expensive and not always affordable to the clinic or the patients, even though its clinical value. Due to all the above, it is currently not widely adopted.
However, a recent review publication stated that the future is in real-time computer-based sperm selection with objective selection and measurement at high magnification. In addition, the paper stated that such a system has the possibility to eliminate the differences among ICSI operators in terms of fertilization rates and embryo development rates, and, more importantly, the possibility of improvement of clinical outcomes and reduced birth defects. At BAIBYS, we bring this future now. Interestingly, 3 years before this publication, we started developing exactly such a system. The BAIBYS system is a fully autonomous, simple-to-operate system for sperm selection at high magnification. Just place a cartridge with the sperm sample, select how many cells are required, and press start. A few minutes later, when the system beeps, take the cartridge out with the requested number of ideal cells isolated in a separate target droplet. As shown here, the system includes a reusable console and disposable cartridges. Once the process starts, the system autonomously scans the sperm sample, identifies ideal cells based on morphology at high magnification and motility, and robotically transfers them into the target droplet. In contrast to the manual selection that was discussed earlier, the BAIBYS system is fast, a few minutes instead of a few hours, consistent, objective, and even more accurate than the human eye due to modern high-resolution digital cameras. Fully automated, no one needs to be even near the system during its operation, and affordable to both clinics and couples. Therefore, we see its use in every ICSI cycle. Our system is based on AI in several levels. Multiple studies have shown that AI is capable of analyzing sperm cells and especially their morphology based on various scanning technologies. To train our AI algorithms, we recorded hundreds of hours of video of sperm cell at high magnifications. We captured tens of thousands of images, each of them containing a few sperm cells. We asked highly trained embryologists from different countries to label these cells, at least 2 labelers for each cell, each blinded to the labeling of the others. As you can see on this slide, the classification instructions for the labelers were taken directly from the WHO manual in blue, with additional instructions for the BAIBYS labeling tool in green. At the end of the labeling process of each cell, it was classified as either normal, abnormal, in a side view, too blurry to classify, or an unknown classification. The classification for each cell was set only if there was a consensus among the embryologists who labeled it. Otherwise, the specific cell was not taken into account for training our AI algorithms. We have 2 main AI algorithms. The first algorithm was trained to autonomously identify sperm cells in the live video of the sample and to ignore all other objects in the image, even if they appear similar to the cell. However, the core AI algorithm is the one that classifies all multi-cells in real time. This algorithm classifies each cell as either normal, potentially good, or abnormal. A cell is classified as abnormal and marked in red as soon as any morphological defect is detected in the cell. If a cell without a morphological defect is detected, it is classified as potentially good and marked in yellow. The system then brings this cell to the center of the field of view and starts tracking it robotically to gather more images of it while automatically keeping it in focus. After tracking the cell for a second or two, when a sufficient number of images of that cell without defects were collected, the cell is classified as normal and it is candidate for extraction. Let's see it in action. Here is the system tracking a cell that was classified as normal. Above it, a cell with a vacuole, which was therefore classified as abnormal. Here is another cell that was detected without morphological defects, so the system keeps it in the center of the field of view while keeping it in focus until enough views confirm that it is indeed normal. Please note that the system is not confused and does not lose track even when cells cross each other. In addition, the BAIBYS system evaluates various motility parameters of all cells. A sperm cell will not be extracted to the target droplet if its parameters are abnormal. Once a cell is classified as normal based on both morphology and motility, the robotic system starts extracting it. We did not invent anything new here. The robot simply replicates the actions of human embryologists using a micromanipulator, a standard off-the-shelf glass pipette, and a pump. The robot brings the cell near the pipette and extracts the cell into it with the pump. Then the pipette is robotically moved to the target droplet and the cell is relocated there. The process repeats until the requested number of cells was relocated to the target droplet.
So let's see a full cycle of the entire system in action. The embryologists take the sperm sample after preparation using density gradient or swim-up method or microfluidic method as usually performed in your own laboratory. The sperm sample is then placed in the designated droplet in the BAIBYS cartridge. The cartridge is then placed into the BAIBYS console. The user selects the number of sperm to be extracted and then press start. The cartridge is then moved to the low magnification objective for mapping the drops in the cartridge. Then the cartridge is moved to the high magnification objective for selecting the ideal sperm using our unique algorithm. When the ideal sperm is located by the algorithm, the ICSI pipette is then automatically lowered, the pump is activated, and the selected sperm is aspirated into the pipette. The robotic arm is then remove the pipette to the designated droplet and place the sperm there. The cycle will repeat itself until the amount of requested cells is collected. The algorithm will not activate the pump if there is another sperm in the area. The cartridge is then taken out from the baby console and moved to the standard ICSI station for fertilization.
Our next steps are clinical studies, regulatory clearances, and commercial sales. We are currently running various clinical studies, both internally and in various clinics in Europe. A CE mark for sales in Europe is expected still this year, and soon after commercial sales. In parallel, we will run studies in the US in 2025 based on a protocol that was agreed with the US FDA. What's next? Our vision is to take sperm selection to the next level. Currently, the WHO criteria for sperm selection are man-made. While these criteria are validated, it could very well be that there are additional, perhaps superior criteria that humans cannot detect. Based on the big data that will be collected on individual sperm cells and their clinical results 9 months later, BAIBYS will be able, within a couple of years, to refine the criteria and even suggest additional ones by retraining the classification algorithms on this big data. In addition, a new software, the system will be able to autonomously extract sperm cells in both TESE sperm cases and severe azoospermia cases. To conclude, our product is based on solid clinical foundations, simple, fast, and affordable, with clinical and business value. We were recognized by distinguished organizations such as the Israel Innovation Authority, which provided us with the maximum grant possible for a company of our size, IBM, which invited us to join their accelerator, and NVIDIA, which invited us to join their Inception program. Last, we hit major milestones in completing working system, starting clinical studies, and completing 2 investment rounds, the first by fertility doctors and clinic owners. Together with you, we will allow more couples to have babies. Thank you very much.